Good afternoon again. That's my second chance to be here in that podium and speak on behalf of my other company that's called INEA Advanced Technology. That's what I do day in and day out. Being an industrial scientist, unfortunately, limits your opportunity to report the original research, and I wish I had a chance to include more original data, but we're bound by confidentiality agreements, and I decided today to present an overview of a very exciting area where I've been involved for many years. It's silica-based material for drug delivery, and I thought it's fitting just to talk about silica-based material being in Dubai and surrounded by sand, which is nothing more than silicon dioxide. I put together this person have a different kind of silicas in our lab, and we have things like our silica gels, diatomaceous earths, and things like that. All this kind of silica, they're amorphous silicas, and we have dozens and dozens of different brands, and they all differentiate by three main attributes: shape, size, and porosity. So you can have like spherical particles, you can have branch particles, smaller, bigger, and also porous particles and non-porous particles. I think as drug formulators, we're typically dealing with this two, spherical and non-spherical. Fumed silica, you're familiar with, we use it for gels and semi-solids. And spherical silica, we often use it for solid dose formulations. Okay, fumed silica, usually obtained by flame process. You're simply burning silica chloride with oxygen. So what happens in the end is very branch structures. So when we build viscosity, you need the structures because they simply, branch. They don't just stick together very well. And this is an electron microscopy picture of this. These are three major pathways we use for producing our silicon oxide material. So for silica gel and precipitated silica, you simply take a sodium silicate solution and go ahead with hydrolysis. So depending on different condition at different pH, you can produce different, more or less hydrated products. Then you filter that, dehydrate, and you end up with silica gel and what's also called precipitated silica. So this process is mainly used for industry to produce a variety of silicas. We use in myriads of different products, like tires, fillers. That's number one. What we're dealing more frequently in the lab is very famous salt gel process that start off with very cheap, very affordable material called TEOS, tetraethyl silicon oxide, and depending on basic or acidic hydrolysis, you can produce different materials. So most of the drug delivery materials are obtained by salt gel process. And also, probably the most exciting area that people start developing in the last 10 years is what's called methoporous silica. So it turns out that if you run salt gel process in a certain condition, typical in presence of surfactants, you can produce very, very small 2 to 50 nanometers particles with very organized structure and very, very small pores. And that's part of my talk. We'll concentrate on that. So if we think about drug delivery, one of the first questions you ask, what do we know about toxicity of the silica? Of course, the sand is not toxic. We know that. But what about other forms? So we have plenty of other forms of silica in nature. For example, the shell of the rice is composed heavily about silicon dioxide. Big problem in agriculture, what to do with that. Another example, diatomaceous earth. This is like our algae, small algae, the shells of which is constructed of our silicon material. Big problem, dust. But we use it in the lab. So we know that when we use silica orally, it's pretty safe. We can take up to 5 grams per kilogram, and silica can be found in many orally taken product. But when we start talking about very small particles, I think we're much more concerned because when we use smaller particles, the solubility goes up. So and we need to look more closely in this issue. Again, crystalline silica is pretty much insoluble. So even in high temperature, we cannot reach more than 1 ppm at 400 degrees Celsius. However, the amorphous silica is pretty soluble, especially at high pH. pH are more than 9. But even at neutral pH, the solubility is pretty high. If the particle size is small, we're talking about nanoporous silica, it's 70 ppm, and porous is 120 m. And the mechanism, of course, silicon dioxide is not soluble by itself. So the mechanism, the rationale for dissolution is a conversion, the hydrolysis of silicon dioxide in salicylic acid. And what's known is excreted with urine at about 2 milligrams per kilogram a day. Anyway, I'd like to talk now about mesoporous silica material. That's very, very exciting material, and there's a number of very interesting articles published recently. As I mentioned, this is electron microscopy picture. You have like a honeycomb, a very, very organized structure with very, very high pore volume. We're talking about cubic centimeter per gram. That means that if you have a gram of material, you can load over a gram of other material, for example, oil, and still keeps the silica as a flowable material. I think one of the problems why people don't use it, unfortunately, it has very limited commercial availability. Pretty much every single piece of research is done on homemade silica, and if you check up, for example, the Ulrich catalog, you can find a material like MCM41 for a cost of $563 for 25 grams, which makes it very difficult to do the research. Anyway, so preparation of mesoporous silica nanoparticles. The process has been discovered more than 20 years ago. It's called liquid crystalline template. So it turned out a simple modification to salt gel process can give you these beautiful particles. What you need to do is just bring a surfactant. So when you have a surfactant, you have the micelles. Micelles get aggregated, and when you add tetrahydroxysilicon in solution, they start kind of wrapping up these micelles, right? And then in the end, you have this organized structure. The only problem here, you need just to remove the surfactant at the end of the process. Then you can do like either wash out, or people do the calcination, and you end up with this kind of particle. So what can you do with this particle? Of course, when you have a silicon oxide particle, you have plenty of hydroxy group sticking on the surface. Okay? So two things people do for drug delivery. They use either endogenous hydroxy particles, or they do surface treatment of silicon with other silicon material. So typical people use amino-modified silica. And you end up with all the chemistry done either with hydroxy group or with amino group. So that's pretty much all the things you see in the literature for conjugation. So the question is, compared to other drug polymer candidate, what's the potential of that thing? We've seen a couple of talks about drug polymer conjugates. Can this system match that? Can this materials be produced economically? What's the density of conjugation? Okay. Now can I talk a little bit more about how you do surface functionalization? That's a key for drug delivery. And here you have our two options. The options number one, you build surface modified material in one pot. That means you take our tetrahydroxysilicate, add other silicon-based material with organic group, and do everything in one pot process. It's very versatile, very convenient, but one of the biggest problems with that, you might have a pore clogging, because it's not necessary you have a homogeneous distribution of things, and the organic part will start just clogging the pore. The second option, it's called grafting method. So we have our homogeneous distribution, but unfortunately in this case, you need to produce silicon particles, and then as a second process, use hydroxy group and do other silicon-based chemistry. Okay, now, how that potentially match our other drug delivery modalities. I think on that picture, there's like probably 6 key modalities, and today we've heard about, I think, few of them. We've heard about liposomes. We've heard about inorganic RNA particles from you. I don't think we heard about virus-like particles. That's more exotic part. So definitely melanoporous silica belong into this class. So what options do we have? What people try to conceptually show that? And I think the possibilities are really exciting because what you can do, you can load the drug inside of the spores. You can decorate the surface of the particle with whatever targeted moiety, and then you can potentially clog the pores with temporary corks and then open them up. Okay? So what are the problems? Unfortunately, the silica surface is relatively hydrophilic. You have a lot of age groups. So when you try to load drugs, and a lot of them are hydrophobic, it's very difficult. So the loads level are relatively low. So how to overcome that? You need hydrophobic surface modification. So people were working a lot in how to make the surface more hydrophobic so that you can just put more drug, increase the loads. Okay? This is like a conceptual thing, what you can do with mesoporous silicon nanoparticles. And I think the exciting opportunities is that you have 3 different options. Having a pore, you can decorate the pore, you can decorate silicon core, and you can run a chemistry in such a way that only the surface of the particle is decorated with target marker. So by combining these approaches, you can have a very, very powerful tool. And there are very many examples in the literature, but I decided in the interest of time, I'm going to present you only one, which I found like very elegant. I don't think it's ready for moving in clinical trials or anything like that, but it just illustrates the power of this approach based on mesoporous silicon nanoparticles. And there was a series of articles published by Spanish and U.S. investigators. This is the thing is from the article from Mary Valea Reggi in Spain, published a couple of years ago, and that had like a very, very interesting example for detection of DNA molecules. And DNA, for example, could be originated from viruses, from microbes. So it's essential for any environmental detection or biological threat. So if you put a catalyze on the surface of the particle and put a little bit of hydrogen peroxide, this particle starts moving around because you produce oxygen bubbles. Okay? So that's one part of the molecule, so this one. Then this is the analyte you try to detect. This is a very small particle, less than 500 nanometers. You don't see that through the microscope. But then you have a complementary big particles decorated without a DNA molecule. So this analyte DNA is complementary to this end and this end of the molecule. So if the detection is right, this DNA molecule binding this and this particle. Now, this big particle that are moving randomly in absence of the analyte, then connected to this small particle that's moving by catalyzed activity. And if detection is taking place, all the thing is that just moving not a random fashion, but in some kind of like organized fashion. Therefore, without any complicated tool, you can simply look in the microscope and see the movement of the particle. So I found those like really, really fascinating things. There are many more examples, and I want to go to the closure and ask her, myself, and present this question to the audience. That silica is very, very fascinating, maybe a less known drug delivery, vehicle. It has huge potential because it looks like it just maybe have little toxicity, maybe cheap, versatile, could be commercially successful, and maybe like our liposome. So what we can do, we already know that we can work with poorly soluble drugs, load them. We can use it for imaging. We can use it for targeted delivery. The area that's very, very little explored, and I'm curious about that, very little people try to do biologic. and you can have very, very fascinating stimulate turbulent delivery. So challenges, potential toxicity. Nobody really studied long-term toxicity. Manufacturing, and I mean industrial manufacturing. We have a potential low drug load, and if we use any kind of smart approaches, of course, as an industrial chemist, I ask myself, can we come up with real manufacturing processes? But anyway, I think we have a nice driving forces that will propel silica to new heights soon, because we have our solid industrial basis. People use silica and develop and process around silica in areas other than pharma, for example, catalysis, inks, polymer. And I think at the end of the day, as one of the early speakers said, we have so many problems, we need new drug delivery system, and that's potential candidate. It's an interesting technology, and if I had a chance to fascinate it a little bit, I think my function was served. Thank you very much. Okay, that's my thank you slide. I mean, I don't think the weather, that's what we have in Boston right now. We have like piles of snow. And I want to thank a couple of guys that working with me. My partner Nelson Landau, Ken Avery, and unfortunately we have an exciting project with a Unilever company in Port Sunlight. And I put the names of the people that were there, very stimulating in discussion, and that brought me an idea just to do this presentation. And I also, if anybody's in the Boston area, I'm a co-chair. We have an industry organization called Massachusetts Biotech Council. So we help to set up a dialogue between industry and academia. So we have a series of seminars on drug delivery, and I'm a co-chair of this organization. If you have any interest, if you're passing through Boston, let me know. I'll be happy just to moderate presentation and interaction with the colleagues that are based in around Boston area. Thank you very much. Do you have time for a few questions?
Maybe one or two questions. Yes.
No, this nanoparticle, this is a single, if we're talking about mesoporous nanosilica, no, this is like round, non-aggregated particle. That's the beauty of it. You saw like microscopic picture there, they're not aggregated. They're singular, separated particles with very, very organized pore structure. You can do it yourself.
One question.
Yes. I think, sure. I think what I do, every day, I do mostly solid oral dose, and we use silica. I don't think we have any concerns about, using silica in solid oral doses. I think we feel very safe. It's a grass material according to our, FDA, and I try to remember whether we have any limits on the amount of our silicon oxide and the tablets. We might have some, but I can't recall any single project, and we oftentimes put silica for different reasons in solid dot formulation. I think it works pretty well, and lately, what people promote, especially for example, Grace Company, one of the biggest producers of silicon oxide, what people interested, the precipitated silica for drug release are not very good because it's not very porous. There's few particular grades, if you're interested, that are much more porous. And then they advocate, if you're interested, just to put the drug, to load the drug for release, There are a couple of, or probably more than a couple, but there are a handful of particular grade that the company promotes very heavily. They are FDA approved, and I've seen a lot of fresh material there. They suggest, for example, this is the best way just to load oily material in your tablet, precipitate on the silica. I think what people are a little bit uncomfortable, it's not about the toxicity. People are very, very particular about the kinetics of release. So sometimes, especially for very hydrophobic molecule, people are concerned you put it in the water, right? So what will be the driving force of the release? It's like monophasic, biphasic. I think it's major concern, not toxicity. And I think small particles. This is the future. But I think people try to use more silica, and I think we have green light here.